Today I want to discuss about the introduction to molecular biology. Before going to the molecular biology, first I want to explain about the definition. Well, coming to the definition of molecular biology is the science that studies all living processes in cell at the molecular level. So this is the definition of molecular biology. Well, coming to the some historic events in the molecular biology. The term molecular biology was introduced by William. So here in the year 1928, Griffith discovered a phenomena, the transformation. So this is the first experimental evidence to support DNA as real. According to the transformation experiment which was conducted by the Griffith in the year 1928, he selected two different strains of bacterial cells which belongs to the diplococcal pneumonia. So the first strain, it is called S strain. S, that is nothing but smooth strain. And the second one that is called rough strain, that is he denoted it as a R strain. So here the S, which means the smooth, and as well as the main difference in between these two strains, which is once if you cultivate these bacterial cells on the agar plate, the S strain will appears as a smooth appearance, or it will produce some mucus layer on the top of that particular colony due to the secretion of a polymer, glucuronic acid. So due to presence of these polymer, it appears as a smooth colony. In addition to that, it is a virulent one, which means it causes disease, or it appears, or when if it enters into an organism, it shows some disease symptoms. That's why this is called the virulent strain. And the second one, that is called R strain. So here the R strain is failed to produce the polymer on the colony. So colony, that's why it appears as a dull or rough the physical appearance of the colony. It is the non-virulent because it cannot cause any disease symptoms if it enters into an organism. So here he selected the S and R strain. Randomly he selected some mice. So here in the first attempt, he injected the S strain to a mice, and he observed that particular mice. So here that mice was died because of the pneumonia symptoms. So here in the second attempt, he injected the live R bacterial cell to the another mice. The mice alive it could not express any disease symptoms. So that's why this is called the hybrid mice. In that particular mice, he injected, yes, it failed to express its characters on the experimental animal, that is the mice. So it is also alive. So in the fourth attempt, he injected the mixed version of the cells that the mixed version of substances. The one is called R bacterial cells and the next one is called heat killed. So by the combination of these two, so that is called in the second and the third mice, generally they are alive. So in the fourth mice he injected R and as well as the heat killed S. So here in the he injected that particular uh, substance to the fourth one, so the mice will die and it expresses some specific that pneumonial symptoms. He concluded that the DNA is the responsible substance to carry out the information from one generation to the another generation. So here there is another experiment. It was conducted by Avery, MacLeod and Carty. That is the continuation for the transformation experiment, that is called the transforming principle. So he named to that particular substance, or here in this experiment they have selected, instead of the direct bacterial cells, they have isolated the DNA from the bacterial cell. They have isolated both DNA from both S and the smooth strains and as well as the rough strains. So here in their experiment, they have treated that particular transforming principle with different enzymes, that is called proteases, RNases and the DNAs. For example, if the transforming principle, or that is called the inherent substance, if it belongs to the protein, automatically the proteases will act, and in the subsequent reactions or in the subsequent observations, generally they fail to produce S colonies. So here in their attempt, the transforming principle which is distributed into the three tubes, the first tube they have treated with the proteases, and the second tube they have treated with the RNases, and the third one they have treated with the DNases. And they have, in the first one there is no proteins, and the second tube there is no RNA, and the third one there is no DNA. So here they have cultivated, they have added this transforming principle along with the live R bacterial cells. So here in the first and second tubes, they have observed the growth of S strains along with the R. So the number may be less, but there is a appearance of S type strains. So whereas in the third tube, they fail to produce S strains, even a single smooth strain which was not developed in the third one. So as per the observation, or according to that particular observation, the DNA is the responsible substance to carry out the genetic information from the one generation to the another generation. So there is another experiment is there, that is called Blender's experiment, popularly known as Blender's experiments, which was conducted by Hershey and Chase. So according to for this a bacteriophages. So we all aware about the bacteriophages. The bacteriophage thing, but it can commonly known as a bacteriophages. So they have selected T4 bacteriophage. So here the T4 bacteriophage, it is having a simple structure. They contain protein code. Within the protein code they contain the DNA or the genetic material. So which means we have the two different components. One is called protein and the second one is called the DNA. So DNA they have treated with different types of isotopes. The one is phosphorus, the radioactive phosphorus, and the second one the radioactive sulfur. So here the sulfur, that is called the sulfur 35, and whereas in the case of phosphorus, that is called phosphorus 32. So here the sulfur 35 is used to label the proteins, and the phosphorus 32 they use it to code DNA particles, because there is no DNA. That's why they have selected the phosphorus, radioactive phosphorus, to stain or to label the DNA. And whereas in the case of protein code, it doesn't contain any phosphorus substance for the bacteria. The two different biomolecules, the one is the protein which is coated with the sulfur, and the another one the DNA or the genetic material which is present within the T4 phase. They have labeled with the phosphorus 32. So here the two these two labelings which is done to the two different bacteriophages. The one bacteriophage phase coat protein is coated with the sulfur 35, and for the second bacteriophage they have treated with the they infected a bacterial cell or the E. coli cell. So here, if the protein is a genetic material, automatically in the subsequent observations we can find here they have um, infected to the bacterial cells, and in the kitchenware blender they have mixed for the separation. So here after that they have centrifuged these two cells and the lysate cell. So here in their separation, the sulfur moiety, they have the protein ghost. After bacterial infection, the protein, we all know, the protein ghost, it remains outside the cell. That's why automatically they are less than the bacterial cells. Automatically that is present within the supernatant. So here they have observed that the sulfur moiety that is present within the supernatant, and as well as in the pellet that contains phosphorus. So here after separation, he infected to uh, the new generation of cells. So here in that particular generation, they observed that the presence of phosphorus 32, but there is no presence of sulfur 34. So on the basis of these observation, they have concluded as that is the clear evidence. This experiment is the one of the clear evidence for the to prove the DNA as the genetic material. So here we have the another experiment that is called the conjugation experiment. So here in the bacterial conjugation, it was conducted by the Lederberg and Tad. So that is called the conjugation Here, these are the four different types of nitrogen bases that is present within the DNA. So DNA, that which is made up of nucleotides, we all know about the DNA. The DNA, it is made up of nucleotides. So here, apart from these nitrogen bases, the DNA that contains the nucleotides that contains sugar moiety, that is which is made up of deoxyribose, pentose sugar, it is a pentose sugar, and the phosphate backbone, and the four nitrogen bases. Broadly, they are divided into two different groups. One is called purines, and the next one is called the pyrimidines. So in the DNA, we have two different types in the guanine. These two are double-ringed structures. They are called purines, and the two single-ringed structures, they are called thymine and the cytosine. Generally, that they are called pyrimidines. That is present within the DNA. So here in the RNA, instead of thymine, it contains uracil. So this is the about the chemical composition. So with this, I'll conclude. The next lecture we will discuss about the remaining things. Thank you.